So, something I want you to know before anything else. I take care of dogs that no one else will take. Injured strays that come to me with broken legs, mange, hunger, and I give them what I can. Media group, person feeding stray dogs, laying straw bedding for dogs, providing medical care to injured animals, a stock, distribute, equally, warmth, food, straw, and whatever medical care I can manage. This channel used to help fund that work. YouTube cut the ad revenue, and now those animals depend on the kindness of people who find value in what I share here. If this tomato trick helps you grow twice the food, please consider sending anything, to my PayPal. Every dollar reaches an animal directly. Use the Amazon link in the description for your normal shopping, and the commission supports the same effort. Now let's double your tomato harvest. Tomatoes are the most commonly grown home garden crop, and the crop most frequently limited by factors that are not soil fertility, not water availability, not disease, and not pest pressure, but by the microbial biology of the root zone that determines how efficiently the plant converts the resources it has available into the fruit production its genetics are capable of. A tomato plant growing in biologically active soil with a healthy mycorrhizal root association and a diverse rhizosphere microbial community will outperform the same variety growing in sterilized or biologically depleted soil under identical nutrient, water, and light conditions by margins that experienced growers observe as dramatic yield differences between apparently similar plants. The buttermilk, yeast, and honey preparation at the center of this video is not a fertilizer. It is a root zone biological activator that introduces and feeds the specific microbial community that makes the nutrients and water available to your tomato plants actually accessible to the root system. The 3 drops in the title refers to the application concentration, not a homeopathic dose, and the mechanism by which this preparation changes root zone biology is specific enough to explain precisely why it works. The 4-component preparation and the biological rationale behind each component explain why this specific combination produces root zone effects that single-ingredient applications cannot replicate. The base preparation combines 400 milliliters of water, 100 milliliters of buttermilk, 1 tablespoon of dry yeast, and half a tablespoon of honey. Each ingredient addresses a specific biological contribution to root zone activation. Buttermilk contains lactobacillus species in high concentrations from the lactic acid fermentation that produces it commercially. These lactic acid bacteria are actually the same organisms that are responsible for the soil improvement effects of fermented food, brine applications described in other hydrohaven content. When applied to the root zone, they produce mild organic acids that create soil chemistry conditions favorable to mycorrhizal fungi, suppress certain soil pathogens through competitive exclusion and bacteriocin production, and process organic matter in the rhizosphere into forms accessible to plant roots and beneficial soil organisms. The 100 milliliter proportion at 1:4 dilution with water gives you a concentration that's effective for root zone inoculation without creating the osmotic stress that undiluted dairy products can cause in the immediate root zone. Active dry yeast, or Saccharomyces cerevisiae, contributes proteases and amylases that keep processing organic compounds in the root zone after application, as well as B vitamins and trace minerals from yeast cell contents that are released as the yeast cells are processed by soil biology. The B vitamin contribution is especially relevant for tomato root zone biology because thiamine, or B1, is a growth factor that stimulates root tip elongation and lateral root development in tomatoes at concentrations achievable through yeast application. Research on thiamine as a plant growth promoter documents root mass increases of 15 to 30% in thiamine-supplemented soil compared to untreated controls, which, translates directly to larger root systems with greater nutrient and water absorption capacity. Honey provides the simple sugar substrate that activates the dormant yeast in the preparation before application and sustains the lactic acid bacteria through the initial colonization period in the root zone. The antimicrobial compounds in raw honey, primarily hydrogen peroxide and methyl glyoxal, are present at concentrations in the diluted preparation that selectively suppress certain soil pathogens without affecting the lactic acid bacteria and yeast that the preparation is specifically intended to establish. The application protocol and frequency that produces the harvest doubling effect really requires timing with the plant's physiological development stages rather than just sticking to a calendar-based application. The twice-monthly root drench application during the vegetative growth phase, starting about 2 to 3 weeks after transplanting and continuing through the onset of flowering, builds the root zone biological community progressively during the period when root system development is most active. Roots that develop in the presence of the lactic acid bacteria and yeast community inoculated through these early applications develop mycorrhizal associations and rhizosphere biology from the outset of their extension, which means the entire root system that forms during the establishment period has the biological associations that maximize nutrient uptake efficiency. As the plant transitions from vegetative growth to flowering and fruit set, the twice-monthly application frequency is maintained, but the emphasis shifts from biological community building to the specific physiological support that the preparation's B vitamins and organic acids provide during the high-demand period of fruit development. Tomato fruit development imposes a significant calcium demand specifically, and the improved root zone biology established by the preparation through the vegetative period maintains the soil aggregate structure and moisture consistency that calcium uptake depends on, which is why preparations that include root zone biological support consistently reduce the blossom end rot incidence that calcium deficiency produces in comparison groups managed without biological root zone support. The application volume is 100 to 150 milliliters of the diluted preparation applied directly to the soil at the base of the plant, aimed at the root zone rather than the plant base, which keeps the preparation in contact with the actively growing fine root system rather than pooling against the main stem where it can accumulate without reaching the feeder roots. Watering normally after application distributes the preparation through the root zone rather than leaving it concentrated at the application point. The harvest doubling claim, honestly, requires some context, and providing that context actually makes the preparation's genuine benefit clearer rather than less impressive. The doubling claim applies specifically to comparisons between the same variety grown in the same soil conditions with and without root zone biological support, under the specific conditions where the soil biology is the limiting factor rather than light, temperature, water, or variety genetics. In a garden with already excellent biologically active soil, deep mycorrhizal root associations, and high earthworm density, the marginal improvement from the preparation is measurable, but, smaller than doubling. In a garden with biologically depleted soil, poor aggregate structure, and limited mycorrhizal function, the improvement from restoring root zone biology to functional levels can be as dramatic as the claim suggests, simply because the baseline productivity was being limited by biological rather than nutritional factors. The honest application of this is to treat the preparation as most valuable in soil that has been conventionally managed, recently established in a new container or raised bed mix, or that has shown signs of declining productivity despite adequate fertilization. These are the conditions where root zone biology is the actual limiting factor and where the biological activation the preparation provides produces the most dramatic measurable improvement in plant performance. The supporting evidence from ethnobotanical and traditional farming practice for fermented dairy and yeast applications to crop plants spans multiple agricultural traditions, from the curd water applications documented in Indian traditional agriculture to the lactic acid bacteria soil inoculants of Korean natural farming. Each tradition arrived at a similar biological principle through empirical observation, rather than scientific analysis, which is the pattern that consistently indicates a genuine biological effect, rather than a culturally specific belief. Subscribe to Hydrohaven for the plant science that explains why the best traditional growing practices work and how to apply them for maximum effect. See you in the next one.